thanks to everyone for a remarkable outpouring of generosity and support for the university in this challenging time. We're incredibly grateful, and we have a terrific group of scientists who are continuing important work during this period. What I'd like to do now is go to the slides. So this is a picture of a typical coronavirus, and this is a virus that is much smaller than any cell. It contains on its interior a single large RNA molecule that encodes all of the genetic information required for replicating the virus. It's enclosed in an envelope that includes these big purple spike proteins, which are the proteins that engage a receptor on every cell that gets infected, by which it gains entry into the cell. So the life cycle of the virus is pretty straightforward. It attaches to a specific receptor called the ACE2 receptor on particular cells that express this protein at the cell surface. Through this receptor, it gains entry to the cell. It then is uncoated, allowing the RNA, the genetic material it contains, to be translated into new proteins that then get cleaved into particular proteins that partake in the replication of the virus, the production of all of the proteins required for assembly of the virus, and eventually new virus is shed from the cells that are thereby infected. So this is a new virus that has not previously existed in the population. It's believed that, like other coronaviruses, have gone on to cause serious infections in humans in the last 20 years. It started in bat species, and these bats, either through intermediate hosts such as civets or camels, ultimately acquire the ability to infect humans, and there then becomes human-to-human spread, accounting for the disease that we see spreading through the human community now. So, like other viruses, it has ability to spread from human to human, and this R value is the number of individuals who will typically get infected from a single affected individual. So the reproductive rate in China, before any recognition of the epidemic was achieved, showed evidence in retrospect of a reproduction rate of about 4. Meaning that every individual who is infected passes the virus on to 4 new individuals. And so as a consequence, you can see the ability for explosive growth, because you go from 1 person to 4 to 16 to 64 to 256 to 1,000, and you're off to the races. So in order to control the virus, you either need the ability to stop the transmission by a vaccine, by medication, or by simply isolating individuals and preventing them from encountering other people who they will then infect. So the typical course of infection with the virus SARS-CoV-2, which causes the disease COVID-19, it typically incubates for about 5 days from the time of exposure, with a range of 2 to 14 days. Typically causes fever, dry cough, and fatigue. Some people have minimal symptoms and may not be aware that they have been infected. About 80% of infected people recover without complication. 20% need to be hospitalized, typically due to progressive shortness of breath. And interestingly, this doesn't usually start until days 5 to 9 after the beginning of symptoms. About 5% of infected people require care in an intensive care unit, and many of these require intubation, going on a ventilator to assist with oxygenation of the blood. And in most societies, 2 to 4% of all infected people will end up dying from this infection, which is a very high number for a respiratory virus. So in New York, this is data from the current epidemic in New York, and it makes the important observation that the death rate accelerates with age. Under age 17, it's exceedingly rare for people to die from infection with the virus. It's still very rare, about 1 in 400 individuals under age 45. Between 45 and 64, it goes up to 1.5%. Goes up further between 65 and 74 to a little over 4%. and over age 75 is about 11.5% of people will die from the infection. There are particular risk factors that expose people to adverse outcome. These include diabetes and some other underlying conditions. But next slide, there's one particular feature which is quite striking with this virus that is unusual. And that is a very strong bias toward a greater rate of death in men than in women. Shown at the bottom in solid red is the fraction of infected men who die in different ages, and on the right, the corresponding fraction of women. And at every age, there is a marked discrepancy with men dying much more frequently than women. And we don't understand the reason for this difference at this time. So there's a number of key questions that this is so new in the population, we really don't understand all of the factors that contribute to the biology of this disease. We don't know what explains the late progression of the respiratory symptoms. It's striking that it appears to have its onset at about the same time that the immune system is typically kicking in. And there's strong speculation that it may relate to the activation of the adaptive immune system that Michel Nussenzweig will talk more about. We don't know what explains the marked difference in the outcome among males and females who are infected. Like other viruses, we expect that we may need to treat people early in the course of their disease in order to maximize the benefit of any therapy. And we are not diagnosing most people early in their course. We're typically diagnosing them now when they present to the hospital because of respiratory symptoms or other serious medical problems. And as a consequence, we're not seeing people early in their infection, most of whom are staying at home and not even getting tested. We don't know whether recovered patients are immune to reinfection, and if so, for how long. We are just learning from Michel's work and others whether recovered patients have antibodies that can prevent infection in themselves or in other people. We don't have current antivirals that have been designed or optimized for SARS-CoV-2 virus. We've had severe limitations on testing, and as a consequence, we don't know the true number of infected, and we also don't know how frequently people are asymptomatic when they're infected. We will need highly specific serologic testing in order to identify people who have previously been infected, and these antibody tests are just coming online now. So thinking about vaccines, which everybody has put very high hopes into, vaccine candidates can be rapidly produced. However, it is no sure bet that the first vaccines that are produced are going to be either efficacious or safe. There are some vaccines in recent years for diseases like dengue fever and SARS, which is a related virus to the SARS-CoV-2 coronavirus, where it has turned out to be very difficult to make effective antibodies and, in fact, effective vaccines. And in fact, some of the vaccines that have been produced have in fact not only not worked, they have caused harm. It's unlikely that we will have a vaccine available for at least a year. There are some therapies that are currently under investigation and are being used under compassionate use in hospitals. There are several antivirals. One, remdesivir, has received a lot of publicity. It's an inhibitor of the viral RNA polymerase, and it shows efficacy in vitro in cells. You can prevent cells from getting infected by prior treatment with the drug. And these are still in clinical testing and not approved by the FDA. Hydroxychloroquine, an anti-malarial drug, has received a lot of publicity. It is also unclear whether this is going to work as a treatment in patients. Other protease inhibitors that were built for HIV proteases have failed in clinical trials and are no longer being widely used. Because of the unusual acceleration of the respiratory disease relatively late in the course of infection, people have suspected the immune system is playing a role, and use of corticosteroids or antagonists of a specific protein called interleukin-6 have been used under compassionate use and are now in randomized clinical trials to determine whether these can attenuate the acceleration of the respiratory disease that requires so many patients to go on to intubation, and ultimately many of these people are the ones who die from their disease. Other repurposed drugs are being tried. Repurposed drugs are drugs that have been used for other purposes, but by study of their effects in vitro have shown some activity against the SARS-CoV-2 virus. So this shows the daily death rates from COVID-19 in countries around the world. And you can see that in many countries, the disease accelerates, reaches a peak, and then levels off, and in some cases goes down to very low levels. So for example, China had the first exposure to this disease in Wuhan. They went up to about 200 deaths per day, were able to contain the virus, and now have very few deaths from this virus. The US has been steadily going up and is now, in just the last several days, started to show a plateau of number of deaths that are every day from this virus, which currently sits at about 2,000 deaths per day across the country. This shows data for the state of New York from Governor Cuomo's presentation this afternoon. I think the salient point is starting on March 20, 2 days before the state shutdown, stay-at-home order was put in place. There were nearly 500 people being hospitalized every day due to infection with this virus. This went up to a peak several weeks ago of over 3,000 people being hospitalized every day. And we stayed near the plateau for over a week, but over the last week have started to come down very significantly. And so from that peak of over 3,000 cases per day, yesterday there were only 1,300 patients admitted to the hospital for COVID-19 across the state as of yesterday. So this is all headed in the right direction and is quite promising, and is a direct consequence of everybody doing a good job of staying at home and not going out unless absolutely necessary. This has been followed by a lagging indicator, which is the number of deaths across the state, which achieved a peak of about of nearly 800. And you can see over the last 6 days has been coming down rather steadily. Today, again, was at 478, almost identical to what it was yesterday. But this is a marked reduction in the number of deaths every day. So in testing, there are two important types of tests that relate to this infection. The first is a test for RNA from the virus. And this indicates current or very recent infection with the virus. About 20,000 tests are being done every day in New York State. Most people, as I mentioned previously, who are infected do not get tested unless they need to go to the hospital, at which time they're much more likely to get tested. As a consequence, we don't know the true number of cases, only how many people got tested, how many were positive. There's a second test that is rapidly approaching wide use, and this is a test for antibodies to viral proteins, so-called serologic tests, because it's taken from the plasma of the blood or the serum of the blood. And these tests can tell you whether your body has made an immune response to infection, and it's a very good marker, if very specific, of prior infection. Importantly, it may indicate near-term immunity-free infection, but that's a major question that we don't have full data on yet at this point. All we know is that these people recovered from the viral infection and they mounted an immune response. We don't know how immune they will be to subsequent infection with the same virus. These serologic tests are just high-quality tests that are highly specific, are expected to be being used widely in the next several weeks at very large numbers. The PCR test has not been available at nearly enough, a high enough level for wide testing. The antibody tests, I anticipate everybody will be able to get in relatively short order if they want one. This simply shows the decline in the percent of positive tests in Manhattan over the course of this pandemic. And I'll just make the point that in the top row On March 26th, there were 1,000 tests done in Manhattan, and almost 50% of them were positive, indicating that there's very prevalent disease in people who are presenting for testing to the suspicion that they might have the virus. That percent positivity has been declining steadily over the ensuing weeks, and as of yesterday, it was down to 21% positive. So from this, you can deduce that the prevalence of infection, of new infection, is going down dramatically, and we're clearly headed in the right direction. And this goes along with the decrease in the number of hospitalizations and the reduced death rate. So one of the major questions that the nation is facing now is reopening the economy. There's been tremendous loss of the number of jobs in the country, economic decline. Is eager to get back to work as soon as possible. And this raises the question, what would the ideal conditions to do this look like? So you would want very low levels of infection in the population. As of yesterday, there were still 25,000 new cases diagnosed every day. This is a very large number, and as I've indicated, this is the tip of the iceberg. There are many more people have the disease diagnosed by a formal test. You would like there to be sufficient testing capability with rapid turnaround to allow quick identification and quarantine of those who are or who become infected. Right now, unless you are in a hospital, the turnaround for tests averaged 4 to 7 days. These are not rapid turnaround tests that allow public health workers to see that people are isolated from their members and others to prevent the spread of infection. We would really need capacity to trace, test, and isolate commonly infected individuals. We are not prepared to do this at any scale at anywhere in the country that I'm aware of, and this is a major that needs to be addressed. As you think about going out, you would still want everyone who can to telecommute as much as possible. You'd want to maintain social distancing, continue wearing masks in public spaces. You would want exceptional efforts to keep public spaces sanitized, particularly in public transit, which is so heavily used here in New York. And you would want workers who have close contact with the public to have appropriate PPE. And of course, you'd like the serologic testing to identify individuals who are likely to be immune from subsequent infection, at least for some period of time. So in contrast to that, we do not have those conditions in most parts of the country, particularly most of the population centers in the United States. And there are several major questions that remain unresolved at present. This shows two different models for what might happen as a consequence of the course of this epidemic. In the orange line is a model in which you would hit a peak sometime in April and then slowly decline down to very low levels, and the peak would never come back again. That could be achieved if there was high herd immunity, that so many people had been infected that the virus can't find new people to infect, or if the virus was highly sensitive to temperature or humidity and went away as the season changed. An alternative model is that the virus will go down to low levels, but then come back as we ease the social restrictions. Everybody comes out at a time when there are still many people who can be infected, and you get a rebound and the rates of infection go back up. There's also an alternative model in which you hit a plateau, but you never come down because you're not doing a good enough job in social distancing and preventing spread of the virus. And you stay at a plateau until everybody gets infected and the virus runs out of steam because it can't find anybody else to infect. If I were to guess from what we see so far, I will guess that we're somewhere between the blue line and the yellow line, that we're going to be able to get the levels down much lower than the peak, but perhaps not down to zero, and that as our patience for being in isolation from one another goes away, we'll come back out and we are likely to have a resurgence of the viral infection cycle, and we'll have this pattern of waning and waxing of viral infection in the population. which I suspect would be very frustrating for everybody. So at Rockefeller, as I mentioned, we've embarked on a number of ambitious efforts to try to understand the biology of the virus and impact therapeutics. And to quickly articulate some of the projects shown on this slide are Barry Kohler at the top and Paul Banache in the middle. And they've been involved in the convalescent plasma project, which is to take plasma from patients who have recovered from infection and find who has high levels of neutralizing antibody that can prevent the virus from infecting cells, and give that plasma back to either people who need it therapeutically or people who are at high risk of becoming infected, such as in nursing homes, frontline healthcare workers, for example. And we've been collaborating with the New York Blood Center and New York City hospitals in order to implement this. And to date, more than 2,000 plasma samples have been taken, and these have been processed by the Blood Center and are being returned to hospitals for infusion back into patients, and clinical trials are really just getting going. Looking for drugs that can be repurposed, and this is Charlie Rice, who won the Lasker Prize for his work leading to the cure of hepatitis C at Rockefeller. And he's screening through drugs that have been marketed and have proved to be safe in large clinical trials to see if they might inhibit viral replication. Shown here is Michel Nussenzweig, who you'll hear from in a minute, working on things that will take a little longer to get into the clinic, but have very strong potential for utility. And this is developing neutralizing monoclonal antibodies, decoy receptors that can prevent the virus from infecting cells, and nanobodies, an unusual type of antibody-related molecule that Michael Rout is trying to develop against the SARS-CoV-2 virus. This shows the decoy strategy that Paul Banache is taking. The receptor for the protein, for the virus, is a protein called ACE2, and that's normally embedded in the membrane of cells. He is making a soluble version of this that can circulate in the plasma and still bind to the virus, and its back end will enable it to be cleared from the circulation by professional immune cells, and thereby prevent it from ever accessing the cells that cause infection and all of the severe consequences. So this is an example of a decoy receptor. We have a number of investigators who are trying to make novel drugs that target viral targets. Shown here is Sean Brady and Tarun Kapoor and Michael O'Donnell, who are targeting viral RNA polymerase as a target, the helicase that's specific for the virus, and novel proteases that the virus makes that humans don't make, and are using high throughput screens for novel inhibitors of viral replication using technology that Charlie Rice has devised. These show some of the molecules that Sean Brady has identified that come from the gut from studying of the microbiome and are closely related to antiviral inhibitors of RNA polymerase. Charlie Rice and Jean-Laurent Casanova are both screening for host proteins that are required for the replication of the SARS-CoV-2 virus. Charlie is using cell-based screens using CRISPR technology to knock out human genes and see which genes are thereby essential for replication of the virus. Jean-Laurent is studying patients who have extreme consequences of viral infection, either the old who ends up intubated in the ICU with infection, which is a very rare event, who might be incapable of combating the virus, or the 85-year-old in a highly infected in a nursing home who remains free of any signs or symptoms of infection. And these individuals can directly identify host proteins that cause adverse or beneficial outcomes of infection. Lastly, Tom Tuschl and Bob Darnell are both developing advanced blood and serum tests for the virus and the proteins to allow us to assess both the background infection rate, but also to identify people who are at low risk of infection, and also enable us to think about how do we reopen society? who's ready to be out in public, who should be quarantined, and try to do this at a scale that would matter at public health level. So concludes my presentation, and I'd now like to turn the podium over to Michel Nussenzweig, who is going to tell us in more detail about his innovative work to develop novel antibody therapies for SARS-CoV-2. Michel?
Okay, thank you, Rick. We've just heard about vaccines and drugs and ways in which we can deal with this infection. And in particular about the plasma therapy. So antibodies, which are in plasma, are the active agents for just about all vaccines. But as we just heard, not all vaccines work. What my lab is trying to do is to find these active agents, the antibodies, in people who actually make good ones in order to reproduce them and be able to give them to people as a substitute for a vaccine. So if I could have the first slide, I'm going to go over a little bit how we do this. So we've done this for a number of different viral diseases, HIV, Zika, dengue, tick-borne encephalitis virus, and hepatitis B. And what we do is we ask for volunteers and look in their plasma for whether or not they've made a superior response to the virus. And what we find in every case we've studied so far is that there are 5 to 10% of people that really stand out, that they're exceptional. And so what we do is we recruit those individuals and begin to look for the antibodies in those people. And I'm going to just show you an example from hepatitis B, and then I'm going to tell you about where we are with this particular virus. So that I can, next slide, please. So what we did for hepatitis B, and this was a collaboration with Charlie Rice, a virologist that you heard about from Rick, who we are continuing to collaborate with for the COVID problem. So we recruited these volunteers, 162 people, and then screened them for the ability of their antibodies in plasma to bind to the virus and also to neutralize the virus. The next slide. So what you see here is the results of that screen. And on the y-axis going up is the neutralizing activity. And on the x-axis is just the ability of the antibodies in plasma to bind. So what you can see is that a lot of people make antibodies that bind, but not too many people produce high amounts of neutralizing activity. And those are the elite neutralizers. So we select those people, and then we clone out their antibodies. And I'll just, on the next slide, is the procedure for how we do this. So we take blood, and in the blood, in the circulation, are the cells that can actually make the antibodies, and they're called B lymphocytes. We identify the ones that actually have antibodies by showing them the viral protein. And that viral protein is labeled in such a way that we can identify the cells that bind to the viral protein. Now we sort them out as single cells and then do the cloning, which is done by molecular biology. So on the next slide, what you see is how these antibodies can be protective. This is a case for hepatitis B. So on the bottom left, so the way this experiment is done is that a mouse is given the antibody and then infected. And if the antibody prevents infection, you'll see that. So on the left is a control where there was no antibody given, and you can see that very, very high titers, logarithmic on the y-axis, they get infected at very, very high levels. If you give the antibodies on the next two panels, you can see that that simply doesn't happen. So that's the idea for prevention, but it also works for therapy for an active infection. So what are we doing for COVID? So next slide, please. So what we've done, and this slide is already out of date, is asked for volunteers, people who have been infected and have recovered. We asked them to have been symptom-free for at least 14 days before they can come here and donate. And to date, the community has been incredibly generous. We've had over 500 calls, and we have scheduled 170 people so far to participate. They come to Rockefeller. They're seen in a single participant room. They get a finger prick to see that their hemoglobin levels are high enough for us to be able to obtain blood from them. And then we collect less than a unit of blood, about half of a unit of blood, which is what you give when you donate blood at the blood bank. And we separate the blood into the plasma, the serum, the cells, and then we screen that for binding and for neutralization. And I'll just show you what we're learning so far. So next slide, please. So this slide shows the neutralization assay for the first 43 volunteers. And the higher you go, the better you are. And you can see that out of these 43, there were really only 11 people that had a sufficient level of neutralization that they could neutralize 50% of the virus in the assay. So not a lot of people are showing high levels of neutralization. And in fact, only 2 of them, or 5%, stand out as exceptional neutralizers. One of them is this person, 21, and the other one, 47. And we were very lucky, next slide, that the first person that walked through the door was 21. And here we are doing the cell sorting on 21. And I'll just show you on the next slide what that looks like. So each one of the dots that you're looking at here is a cell going through a machine called the cell sorter, which helps us identify the B cells that bind to the COVID antigen. The healthy control is on the left, and you can see that there's nothing in that upper square there. And number 21 is on the right, and you can see that there are a lot of black dots there, and those are the cells that we sorted. And further to the right is a pie chart, which shows the 125 antibodies that we've been able to obtain from this person. We've also gotten that number of antibodies from the other person, 47. And we're in the process of beginning to test these, but we already know that some of the antibodies can block the interaction between the virus and its receptor. So next slide. So we're doing this in really an incredible collaborative effort with a lot of laboratories, both here and across the country. And that includes Charlie Rice and Paul Bienash, both virologist experts in viruses. Pamela Bjorkman, who's a structural biologist working at Caltech, which we've had a long-term collaboration with. There are people at the Chan Zuckerberg Initiative, John Pack, who's helping us with proteins, as well as Jim Wells at UCSF, and Wes Van Voris, who is a infectious disease doctor in Seattle. So that's my presentation. I wanted to make it short so there would be time for questions.
Terrific. Thank you so much, Michel. So now to turn to questions. First question to you, Michel, is how should we think about a positive serologic test for SARS-CoV-2? Does that mean that we're immune to reinfection, and if so, for how long?
Okay, that is a very good question. And of course, we don't really know the answer to that. but we can take our thinking from what's known about SARS-1 and other coronaviruses, which is that if you have been infected, you and survived and have antibodies, you're likely to be immune for at least a period of time. So I think that the antibody test, even if you have a low level of antibodies, those antibodies may not protect you, but there are other elements of the immune system, in particular the T lymphocytes, which are likely to be protective. So I would say that in all likelihood, if you have been exposed, you are likely to be immune and will be immune for a period of time, a couple of years.
Great, thank you. So there are coronaviruses out there in bat populations. Do we have any idea how many of those are lurking over the horizon, and can we anticipate what might be coming? Maybe the big question is, can there be a universal coronavirus vaccine, at least for the class of viruses that cause these severe respiratory conditions?
You know, Rick, I don't really know how to answer that question. I don't think anybody does. There's a great diversity in these viruses, as we've heard from the virology experts, and whether or not there are enough shared elements to make a vaccine for all of them is just not clear. I think it's more probable that some of the drugs that are being developed will hit elements that are shared, for example, the RNA polymerase that can be used against multiple different kinds of coronaviruses.
One question that many people had is, what do we know about the patients who are apparently asymptomatic but infected? And maybe I'll take a first swing at that and let Michel follow up. Big problems in this disease is that we have not done wide tests, so that we have not gone into the general population and sampled broad to see who is infected. And so we have very little idea of what fraction of patients who are infected, in fact, have asymptomatic or symptomatic disease. The only systematic study that's been published on this that I'm aware of is one out of Columbia recently, took a series of over 100 consecutive patients who were pregnant, presenting delivery of their newborn child, and sampled all of them and found that 1 in 8 of these individuals in fact had evidence of current or very recent infection by virtue of having a positive RNA test for the virus. So that indicates that there is some reasonable background rate of infection, but we won't know the answer to that until we have more widespread thing in many communities across the country. We really don't know either people are infected but asymptomatic, or do we know how infectious these people are compared to others. Another question that we have is, if you are successful with antibody therapy or prophylaxis, how long would you expect the therapy to last in its efficacy?
Okay, that's a good question. So some of the antibodies that we have can last a very long time. So for example, in the case of HIV, we expect to be able to give the antibodies once every 6 to 9 months. So very relatively long half-lives for antibodies.
Great. So from what you know now, what is your how are you approaching the optimum antibodies? How many people do you think you'll have to study to the single antibody or combination of antibodies to go into clinical trials?
Yeah, so what we did with HIV is to use a combination of antibodies, in fact, 2 that are in the clinic or have been in the clinic a great deal. The idea is to be able to hit the virus in two different locations so that if there is a mutation that allows escape from one of them, then the other one will do the job. And so I think that in this case, to be safe, we may have to do the same kind of thing. And also in this case, it may be that there is synergy. In other words, that 2 is in fact better than additive, better than this plus this, but actually some sort of synergy between the 2. And there's some reports that in fact that's the case.
How long do you think it would take to scale up these antibodies to bring them into use?
Well, for the first type of clinical testing, if we had one today, it would take us about 5 months to get into the clinic. Between that and having it widely available for people is a whole other process, which is very similar, in fact, to what's happening with vaccines. Scaling up a biologic takes time.
Getting back to your extreme patients who had these really robust antibodies, was their clinical course distinctive in any way compared to others?
Well, these 2 particular individuals, they had about 10 days worth of symptoms. But there were others that had 10 days worth of symptoms and were in the lower group. So it's not clear, and at the moment 2 is a small number, so we're really can't say much about that.
One of the aficionados online asked about antibody enhancement and whether you or others have seen any evidence of this phenomenon. You might explain what that is and and tell us what you think.
Yeah. So when antibodies are not potent neutralizers and can bind to the virus, they can allow the virus to go into cells of the immune system that they would perhaps not normally go into and enhance the infection, amplify the infection. And that is a worry, and that has happened, or it does happen in diseases, for example, like dengue. That's the best characterized example of this thing. There is evidence in animal models that this could happen in SARS as well. And so people are concerned about that. They're particularly concerned about a couple of different things. One is the plasma transfers. So if you have a lot of people that don't have very good antibodies, and you give this antibody to somebody that is infected, will it not prevent the disease, but in fact do the opposite? So people are concerned about that. But there is no proof that that is happening in people. And for monoclonal antibody therapies, those things can be avoided by modifying the monoclonal antibody so that it cannot do that.
A number of people have asked about reopening society in several different contexts. Several people have asked, we're thinking about this in the context of Rockefeller University. I'll take a swing at that. As we have thought about opening the university in full, we learned a great deal from our closure. So during the time that we've been closed since March 18th, we had initially a skeleton crew that were our truly essential employees who provided security for the campus, keeping the operations of electricity and power and heat on in the buildings. People who were responsible for maintaining the housing campus. And that was about 85 people from among our employees. We recruited another 150 essential employees in order to provide the support services for these scientists who are working on campus during this time. And so we've had a very active Office of Health Services to monitor these people very closely try to make sure anybody who becomes symptomatic immediately leaves campus if they're on campus. And we've done systematic contact tracing for all of these individuals and have monitored their health closely. And so this has been a bit of a model for us of what we would want to do when the full campus opens up. We would like to know for each person coming on campus, do they have current evidence of infection? Do they have prior exposure with an antibody test? And then follow equally with recurrent testing for the RNA of the virus as an indicator of active viral replication. This is not easy to do, but a small enough community that we think that this may well be possible to achieve. There are several contexts in which it's particularly, several people asked about schools. How are we going to reopen schools? And obviously, particularly in the younger population, social distancing, wearing masks, challenging. And so you can imagine that this will be a particularly difficult environment to maintain. The environment without having of infection. For this reason, most plans for reopening society have said going back to school will not be one of the first things that will be done. But again, you like to have an idea of can you monitor people sufficiently closely to prevent people who have become ill from spreading the virus throughout the population very rapidly. One of the challenges that we will see with this is, unlike influenza, which has an incubation time of a couple of days, the time from exposure to the time that symptoms typically begin is not 2 days like with influenza, but 2 to 14 days with an average of about 5 to 6 days. The SARS-CoV-2 virus. So by the time you're aware that you have the infection, you may have exposed a number of people already. So there are challenges to reopen, and one of the things I think seems obvious is you want there to be a very low level of infection before you fully reopen society. And countries that have been successful in at least partially reopening their societies have had infections across the entire country, typically of 100 per day or less of known new infections. Obviously, at 25,000 per day, we're a long way from that nationally, but there are particular regions of the country or states in the country that have low population densities that have very levels of infection that may be able to open before a big city like New York. So there's another aficionado question here. Is there a second internal receptor that transports the receptor from the endosome lysosome compartment to inside of the cell where it becomes uncoated for replication of viruses? So I'll take a swing at that. So the most interesting evidence in my mind has been evidence of a PI3 kinase 5, a kinase that phosphorylates phosphoinositide at position 5, and so-called PIK5 is involved in endosomal trafficking. And it is not a receptor, to my knowledge, for the virus, but several studies have shown that it's essential for viral replication, and that are inhibitors of PIK5 that in vitro appear to do a good job of inhibiting proliferation of the virus. And a repurposing study, there is a drug that inhibits this enzyme that has been shown in a repurposing study that just came online over the weekend that shows that this target may be a viable target, and there turns out to be a drug that inhibits the enzyme, which may be a drug that is worth trying. And there are hundreds of drugs that are show some activity in these repurposing studies that may be worth looking in detail in clinical trials. One of the biggest challenges that we've had with this epidemic is that the epidemic has hit so fast and so hard in places like New York that although we have thousands of, it has been very difficult to organize randomized controlled trials the way you would like to do. We have much more experience with compassionate use, where people are just given the drug to see if you can help survive without enrolling them in a randomized clinical. Very limited data on what actually works so far. So there's a question about viral load. Do you think the level of exposure to the virus determines the outcome in any quantitative way?
You know, that's true in many viral diseases and bacterial infections as well. I'm not sure it's clear for this one yet, but I wouldn't be surprised if the more you get, the worse off you are.
Yeah, some of the young health workers who have had very outcomes from the disease, and wonder whether people who are exposed to patients at close range when they come in severely ill, their viral load. This is having dictating a particular adverse. Here's another question about vaccines. Is it true that we have never been able to create a vaccine for coronaviruses?
No, there are vaccines for coronaviruses. It's just that, in particular for SARS, there were several candidates that were developed. It's just that by the time they got into testing, SARS was gone, so or very limited, and so it was very difficult to prove their efficacy. And certainly in animal models, there are vaccines for coronaviruses.
Great. So here's a question. Is there any country you feel is doing an exemplary job with coronavirus? I'll say that from out there, Australia and New Zealand, I think, have done quite remarkable jobs of recognizing what was coming in advance, being well prepared for the first cases when they appeared, shut down their society very early, weathered the storm and got down to very low levels of infection, did a good job in the treatment so that the death rates have been extremely low, and have managed to keep their incidence rates extremely low over the last more than a month now. And have never been in the condition that most other countries have been, with very high rates of infection and very frequent outcomes. So I think they stand out as having done a very good job. South Korea is another country that I think has done very well. They got off to a very rough start. You may recall that they had a religious group that did not believe in keeping a distance at all, and had very widespread infection in that population that then spread the infection early in South Korea. And they've done a terrific job of levels of testing to see who's infected, followed them closely, isolated newly infected, and have done a job of maintaining social distancing. And I think they've had a particularly good outcome. And I think, you know, there is, in the absence of effective therapy, the secrets to effective management is recognize what is causing the spread, and that's human-to-human contact, limit the number of people that are being contacting one another, get as many people tested as possible to recognize are there pockets of infection that you need to pay particular attention to prevent spread, isolating people. There's very interesting data out of Wuhan, where the epidemic began, where they started doing test and social isolation and isolating everybody who was infected, but quickly discovered that they're not able to spread the virus. People were being isolated at home because they were infecting all of their family members. And so they only really got to a point where they were able to control the rate of infection when they immediately removed newly diagnosed individuals from their home and put them in these makeshift hospitals or repurposed hotels to enable them to stop the spread. And that was how they got control of the spread of the virus in their population. I see that it's 5 o'clock, so I think it's time for us to draw this to a close. I want to thank Michel for both a fantastic overview of his research, but also for the terrific pace at which his program has taken on and is headed in such promising directions, and for fielding such a wide range of questions so successfully. Thank you so much, Michel.
My pleasure.
I also want to thank all of you who helped in advance to advance this vital research by providing generous support to our special fund for COVID-19 research. We still have a lot of work ahead of us, and your philanthropic support remains vital to our progress. So please see the next slide for more information about how you can contribute and help us combat the COVID-19 pandemic. Thanks again to all of you for being with us today, and I hope you'll join us for future virtual discussions. And until then, all of us at the university hope that you will stay safe and healthy. Thank you and good night.